Hello everyone, and welcome to today's presentation. I'm excited to have you here. Today, we'll be exploring the principles and regulatory aspects in dissolution, with a focus on OEM content, brought to you by Caniska. We'll cover key concepts, regulatory considerations, and best practices to deepen our understanding of this important topic. So let's get started. Let's dive into Slide 2, where we explore the core principles behind dissolution testing. We'll start by looking at the different types of dissolution apparatus commonly used in the industry. Understanding these types is crucial because each is suited for specific formulations and testing requirements. Next, we'll examine the parts that make up these apparatuses, emphasizing which components are vital for accurate and reliable results. We'll also discuss good practices from a compliance perspective, ensuring that our methods meet regulatory standards and maintain quality. A detailed understanding of dissolution operations is essential, so we'll cover the benefits of proper procedures and how they contribute to consistent product performance. Cleaning and validation are key to avoiding contamination and ensuring equipment accuracy, so we'll touch on best practices here as well. Regulatory aspects are always at the forefront. Knowing the guidelines and requirements helps us stay compliant and prepared for audits. Troubleshooting is inevitable, and we'll look at common issues that can arise during dissolution testing and how to resolve them efficiently. Finally, we'll reference the overall index of topics we'll cover, tying everything together to give you a comprehensive understanding of dissolution testing, its importance, and how to execute it effectively. Today, we'll discuss the process by which a solid, such as a tablet or powder, goes into a solution phase, like water. This process is called dissolution. Dissolution testing is used to measure both how much of the drug dissolves and how quickly it dissolves from a dosage form, whether that's a tablet, capsule, or ointment. Understanding this is crucial because the way a drug dissolves directly affects its bioavailability and how effective it will be therapeutically. Now, let's look at the principle behind dissolution. It's all about how a solid turns into a solution, and this transformation impacts how the drug gets absorbed into the body. So, in essence, dissolution is a key step that determines how quickly and completely a medication can act. Keeping this in mind helps us appreciate the importance of proper dissolution testing in drug development and quality control. When selecting a dissolution method, the major criterion should be its ability to discriminate between different formulations by showing changes in the critical attributes that affect the release mechanism. In other words, the method needs to be sensitive enough to detect meaningful differences that could impact how the drug is released in the body. Ideally, the dissolution method used to establish an in vitro in vivo correlation, or IVIVC, should be the same as the one used for routine quality control testing. This consistency helps ensure that the quality control method truly reflects the in vivo performance of the product. It's important to note that a quality control method not based on IVIVC is usually more sensitive to differences in vitro performance, like changes in manufacturing processes. Those differences might not necessarily translate into in vivo effects. Now, regarding the USB tests for finished dosage forms, these can generally be divided into two categories. First, there are drug product quality tests, which include things like assay and content uniformity. These tests help verify the overall quality and consistency of the product. Second, we have drug product performance tests, with dissolution being a prime example. Dissolution testing assesses how the drug releases from its dosage form, which directly relates to its performance in the body. Understanding these distinctions and selecting the appropriate dissolution method based on these criteria are crucial steps in ensuring both product quality and therapeutic efficacy. Let's take a closer look at some of the key applications of this technology. First, you'll notice that these applications highlight how versatile and impactful this innovation can be across different fields. For example, in healthcare, it can improve diagnostics and patient care. In finance, it streamlines data analysis and decision-making processes. It also plays a significant role in manufacturing, enhancing automation and efficiency. What's really exciting is how these applications connect back to our overall theme of advancing technology to solve real-world problems. By understanding these key areas, we see not just the potential, but the practical ways this technology is transforming industries and improving lives. Now, in this slide, we're looking at how we interpret dissolution data. Specifically, we plot the average cumulative amount of drug released, expressed as a percentage against time. This creates what we call a dissolution profile. The key idea here is that this profile helps us understand how quickly and efficiently a drug dissolves over a certain period. By analyzing this curve, we can compare different formulations or test conditions to see which one releases the drug more effectively. This information is crucial because it directly relates to how the drug will behave inside the body, how fast it becomes available for absorption. So, the dissolution profile is a fundamental tool in assessing the performance of pharmaceutical formulations and ensuring consistent, predictable drug delivery. Connecting this to our broader theme, understanding and interpreting these profiles allows us to optimize drug design and improve therapeutic outcomes. Let's take a closer look at the USP 711 dissolution criteria. The key concept here is the quantity, or Q, which refers to the amount of dissolved active ingredient specified in the individual monograph. This is expressed as a percentage of the labeled content of the dosage unit. Now, when we perform dissolution testing, we look at different testing scenarios. For the first, labeled as S1, we test 6 units. In this case, each unit must be at least Q plus 5%. Moving on to S2, we test another 6 units, making a total of 12 when combined with S1. Here, the average of all 12 units must be at least Q, and importantly, no single unit can be less than Q minus 15%. Finally, for S3, we test 12 additional units, totaling 24 units in this phase. The average of all 24 units must be at least Q, but there are additional criteria. No more than 2 units can be below Q minus 15%, and no unit should be below Q minus 25%. This tiered approach ensures that the product consistently releases the active ingredient as expected, which is especially critical for immediate release products. It's about balancing the average performance with individual unit variability to guarantee quality and efficacy. Let's take a closer look at the USP 711 dissolution criteria. This slide outlines the key requirements for evaluating dissolution testing. First, we see that the criteria are structured in levels, starting with level 1, which involves testing 6 units. The main point here is that none of these individual units should have values outside the specified ranges, and at the final test time, no individual value should be less than the designated amount. Moving on to level 2, again, 6 units are tested, but the focus shifts to the average of 12 units, combining the initial 6 with an additional 6. The average must fall within the specified ranges and meet the minimum amount at the final time. Additionally, no more than 10% of these units can be outside the ranges, either above or below, at the final test. Finally, level 3 involves testing a total of 24 units, which include all previous units. The average of these must also stay within the ranges and meet the minimum content. But here, the criteria becomes slightly more flexible. Up to 2 units can be more than 10% outside the range, and no more than 2 units can be more than 10% below the labeled content. Plus, no units should be more than 20% outside or below the specified range or content. This tiered approach ensures a thorough evaluation of dissolution performance, especially for extended-release products. The goal is to balance strict quality standards with realistic variability, ensuring consistent product performance while accommodating some minor deviations. Let's take a closer look at the USP 711 dissolution criteria. The key value here is Q, which is set at 75% dissolved unless the individual monograph specifies otherwise. This means that for most products, we expect at least 75% of the active ingredient to be dissolved. The quantity Q represents the total amount of active ingredient dissolved across both the acid stage and the buffer stage, expressed as a percentage of the labeled content. Now, regarding the testing levels, we have 3: B1, B2, and B3. At B1, we test 6 units, and each must be at least Q plus 5%. So each individual unit needs to meet or exceed that threshold. Moving to B2, we test 6 units again, but here, the focus is on the average of 12 units, combining B1 and B2 being at least Q, and none of these units can be less than Q minus 15%. This ensures consistency across a larger sample. Then, at B3, we test 12 units, making a total of 24 units. The average of all these units must be at least Q, with no more than 2 units falling below Q minus 15%, and importantly, no unit should be less than Q minus 25%. Similarly, there are criteria for the A levels, which are usually more stringent, especially for delayed release products. At A1, with 6 units tested, no individual unit can exceed 10% dissolved. This helps ensure minimal dissolution at early stages. At A2, testing 6 units again, the average of 12 units should be no more than 10% dissolved, and no individual unit should exceed 25%. At the A3 level, with 12 units, the average should stay below 10%, and again, no unit should be above 25%. These criteria are designed to ensure consistent dissolution performance, which is critical for product efficacy and quality. So, whether we're looking at initial release or delayed release products, these standards help us confirm that the dissolution behavior aligns with the intended therapeutic profile. Let's take a closer look at the different types of dissolution apparatuses, as shown in this table. Starting with type I, which uses a paddle apparatus as the USP method, and a basket apparatus for both BP and EP. This highlights how different pharmacopoeias specify different apparatus for the same drug release testing. Moving to type II, we see that the basket apparatus is the USP method, while the paddle apparatus is used in BP and EP. Again, different standards prefer different methods, but both are widely accepted. Type III introduces the reciprocating cylinder for USB, with flow-through cell apparatus listed for BP and EP. This type is useful for testing formulations with particular release characteristics. For type IV, only the flow-through cell apparatus is specified, which is often used for testing solid dispersions or formulations with special requirements. Type V features a paddle over disc setup, providing a different hydrodynamic environment that can be useful for specific formulations. Type VI uses a rotating cylinder, offering another method to evaluate dissolution under controlled conditions. Lastly, type VII employs a reciprocating holder, which is yet another approach for specialized testing needs. Overall, this table illustrates the diversity of apparatuses available, each suited to different formulation types and testing standards. Understanding these differences is crucial for selecting the appropriate method for accurate dissolution testing. Now, let's look at the floating or encapsulated beads method. In this technique, the solution in the vessel is stirred at a specific rotation speed, typically between 50 and 75 RPM. This stirring is achieved using a metallic drive shaft that connects to a cylindrical basket. The rotation of this basket helps keep the beads either floating or encapsulated within the solution, ensuring uniform exposure. This method is often referred to as the rotating basket method, and it's a standard approach within the USP apparatus type 1. The key advantage here is that by rotating the basket, we can effectively simulate the dynamic environment of the GI tract or other biological systems, providing a realistic assessment of the release profile of the formulation. So, the main points to remember are the controlled stirring speed and the use of the rotating basket, which are critical for ensuring consistent and reproducible results. This technique plays a significant role in our overall understanding of how these beads behave under physiological conditions, making it a vital part of our testing toolkit. Now, let's take a closer look at how the rotating shaft is constructed. The shaft itself is fixed to a blade that is attached vertically at the end. This design allows the blade to spin along with the shaft when it rotates. The purpose of this setup is quite straightforward but essential. The blade acts as a stirrer. Its main role is to mix the drug being tested with the liquid inside the holding vessel, ensuring uniform distribution throughout the process. This method, known as the rotating paddle method, is a key part of the USB apparatus Type 2, which we'll see in other parts of this presentation. The design ensures that mixing is efficient and consistent, which is critical for accurate testing. So, in essence, the fixed shaft and attached blade form the core of this apparatus, enabling effective agitation and thorough mixing of the sample. Now, let's look at chewable and ER tablets. One of the key advantages here is that we can perform a multi-pH study within the same test. This approach helps us better mimic how the drug is absorbed as it passes through different parts of the gastrointestinal tract, like the stomach and the intestine. It's especially useful because it allows us to simulate the varying pH environments the drug encounters, providing more realistic data. Additionally, these formulations often require higher agitation and specific hydrodynamics to accurately reflect how they behave in vivo. To achieve this, we typically use the reciprocating cylinder method, which is a part of the USB apparatus Type 3. This setup is designed to provide the necessary agitation and hydrodynamic conditions, making it ideal for testing these types of products effectively. Overall, understanding these methods helps us ensure the proper release and absorption of these complex drug forms, aligning with our goal of developing effective and reliable pharmaceutical products. Now, let's take a closer look at some of the key components involved in the USB apparatus Type 4. First, we have the heated water jacket, which is essential for maintaining a consistent temperature during testing. This helps ensure the accuracy and reproducibility of our results. Next is the tablet holder, designed to securely hold the tablet in place within the apparatus. The tablet itself is, of course, the core sample we're testing, and its proper placement is crucial for reliable dissolution data. Moving on, you'll notice the bead of 5 in diameter. This bead is often used in certain testing methods to simulate the agitation and flow conditions within the apparatus. Now, regarding the configuration options, we have the open loop and closed loop configurations. The open loop allows the dissolution medium to flow through the system, which is simple and effective for many tests. The closed loop, on the other hand, recirculates the medium, providing a more controlled environment for specific testing needs. Finally, the flow-through cell method is another approach used with this apparatus. It involves continuously flowing the medium through a cell containing the sample, which is particularly useful for testing products with complex release profiles. All these elements come together to make the USB apparatus Type 4 a versatile and widely used instrument in dissolution testing. Now, let's look at some key points about the USB apparatus Type 4, which is often referred to as the flow-through cell method. This setup is especially suitable for highly water-soluble drugs. Because these drugs dissolve easily, we can use low volumes of media, which makes the process more efficient and cost-effective. One of the main advantages of this configuration is that it includes an auto-sampler, which automates sample collection, enhancing accuracy and reproducibility. The media reservoir supplies the dissolution media, and then we have the cell and pump unit that circulates the media through the system. This setup is a closed-loop configuration, meaning the media continuously flows through the system without exposing the sample to external contamination. This is particularly important for maintaining consistency and preventing degradation of sensitive compounds. The flow-through cell method involves passing the media directly through a specialized cell containing the sample, allowing for a more uniform and controlled dissolution process. This method is ideal for drugs with high water solubility, ensuring reliable and reproducible results. Overall, the USB apparatus Type 4 offers a modern, efficient approach to dissolution testing, aligning well with the overall theme of improving accuracy and reproducibility in pharmaceutical analysis. Now, moving on to the multi-pH study, it's important to understand that when working with poorly soluble drug products, we need a large amount of media to maintain sink conditions. This ensures that the drug continues to dissolve properly without reaching saturation too quickly. To facilitate this, we use a media selector, which helps us choose the appropriate media for each pH condition. The setup includes a cell and pump unit, which are essential for circulating the media and maintaining the flow during the dissolution process. We also have a splitter or sample collector. This allows us to take samples at different time points without disrupting the system. This entire setup is configured in what we call an open-loop system, meaning the media flows through the cell without recirculating back. Specifically, for this flow, we employ the flow-through cell dissolution method, which is particularly useful for evaluating how the drug dissolves under continuous flow conditions. And as you can see here, we're using the USP apparatus type 4, which is a flow-through cell designed to simulate in vivo conditions more accurately for certain drug products. Overall, this slide highlights how the multi-pH approach combined with a well-designed flow-through system helps us better understand the drug's dissolution behavior across different pH environments, which is crucial for predicting in vivo performance. Now, moving on to slide 17, let's talk about the applications of USP apparatus type 4. You can see it's quite versatile and is used for testing a wide range of dosage forms. For example, injectable suspensions are common, and this apparatus helps ensure their quality. Inhaler drugs, which are critical for respiratory conditions, are also tested using this method to verify their release profiles. Stents and microspheres, often used in targeted drug delivery, can also be evaluated with the USP apparatus type 4. Soft gel capsules and suppositories are other dosage forms where this apparatus plays a key role in quality control. Additionally, active pharmaceutical ingredients, or APIs, and pellets, these small, pelletized forms, are tested to confirm proper dissolution and release. Then, of course, tablets and capsules, which make up the bulk of oral medications, are routinely assessed using this apparatus to ensure consistent performance. Overall, the USP apparatus type 4 provides a flexible and reliable way to simulate the conditions these different drug forms encounter in the body, making it an essential tool in quality assurance across many dosage forms.